Hello everyone, welcome to the channel Microbial World. In this video, we will be discussing about the different types of microorganisms in milk, the source of these microorganisms, and the factors affecting the growth of these microbes. Milk microbiology refers to the study of microorganisms that are present in milk. The milk contains a variety of microorganisms like bacteria, yeast, mold, and viruses. All these microorganisms can either be beneficial, like in fermented dairy products, or it can be harmful, leading to spoilage or foodborne illness. Now let's discuss the main type of microorganisms found in milk. Bacteria is the most significant microorganism that is present in the milk, and it is classified into three categories: beneficial bacteria, pathogenic bacteria, and spoilage bacteria. Lactic acid bacteria is a beneficial bacteria that is very important for the fermentation of milk and also for the production of dairy products like yogurt, cheese, and kefir. These bacteria will convert the milk sugar, that is lactose, into lactic acid, which will lower the pH of the milk and help to preserve it. Examples of lactic acid bacteria includes Lactobacillus species, Streptococcus species, Bifidobacterium species, Leuconostoc species, and Lactococcus species. Pathogenic bacteria can cause foodborne diseases if it is present in contaminated milk or other dairy products. Examples of pathogenic bacteria includes Salmonella species, Escherichia coli, Listeria monocytogenes, Campylobacter jejuni, Staphylococcus aureus, Mycobacterium tuberculosis, Brucella species, and Clostridium botulinum. Spoilage bacteria can cause spoilage in milk and other dairy products by producing off flavors, soreness, and undesirable texture. The spoilage generally occurs in milk when the milk is improperly stored. Examples of spoilage bacteria includes Pseudomonas species, Bacillus species, Lactococcus species, and Clostridium perfringens. Yeast are fungi that can be sometimes found in milk, especially in cases where the milk is not properly stored or when there is high moisture content. While some yeast can be beneficial in case of fermentation, for example yogurt and kefir, some yeast species can lead to spoilage. Examples include Candida species, which can cause spoilage, Saccharomyces cerevisiae. This is used in bread and alcohol production, and it is occasionally found in fermented dairy products. Geotrichum candidum is used in some cheese production, but it can cause spoilage if it is not controlled. Other examples include Rhodotorula species. Molds are also fungi that may grow in milk or other dairy products, especially if the milk is stored in unsanitary conditions. Examples include Penicillium species. It is used in blue cheese production, but it can also cause spoilage if uncontrolled. Aspergillus species can also cause spoilage and it may produce mycotoxins. Other examples include Cladosporium species, Mucor species. Viruses are less common in milk, but it can occasionally be present in case of zoonotic infections, that is, diseases which is transmitted from animals to humans. The milk itself is not a common medium for viral infections, but some viruses can infect the dairy cattle and it can be transmitted to humans through raw milk. Examples of viruses present in milk include bovine viral diarrhea virus, foot and mouth disease virus, avian influenza virus, and hepatitis E virus. Protozoa are single-celled organisms. These are also less common in milk, but it can still pose a risk in case of raw milk contamination. Examples of protozoans found in milk include Toxoplasma gondii, Giardia lamblia, Cryptosporidium parvum. Sources of microorganisms in milk. Microorganisms in milk can come from various sources throughout the production time, handling, and processing stages. One of the major sources of microorganisms in milk is the udder of the cows. Milk from animals naturally contains microorganisms from the udder. These microorganisms can be a part of the normal microbial flora of the animal, but it can also include pathogens or spoilage microorganisms. Infections like mastitis in cows, which is the inflammation of udder tissue. This can cause the presence of harmful bacteria in milk. For example, Staphylococcus aureus, Escherichia coli, or Streptococcus species. This can result in elevated somatic cell count and possible contamination of the milk with pathogens. Milking equipment including machines, hoses, containers, this can also harbor microorganisms if it is not cleaned and sanitized properly. These microbes can contaminate the milk during the milking process. Bacteria can form biofilms on the equipment surfaces. This is difficult to remove and it can continuously contaminate the milk. Human handling can also lead to contamination. This is because poor hygiene practices by the milk handlers can introduce microbes into the milk. Hands, clothing, even the skin of the milker can transfer harmful bacteria into the milk if they come in contact with dirty surfaces or animals. Environmental contamination. Microorganisms from the surrounding environment, such as dust, flies, and airborne bacteria, can contaminate the milk during milking or storage time. Improper storage. This means if the milk is not kept at a correct temperature, that is below 4 degrees Celsius or 40 degrees Fahrenheit, it can result in the growth of spoilage bacteria and pathogens can occur rapidly. Microorganisms like Bacillus, Pseudomonas, and Lactobacillus can thrive in warm environment and result in the souring or spoilage of milk. Inadequate pasteurization. If the milk is pasteurized at a too low temperature or for short time, harmful microorganisms like Listeria or E. coli can survive in the milk and continue to proliferate. Contamination can also occur during packaging time. That is, the packaging materials or the packaging process itself can introduce microorganisms if it is not done in sterile conditions. Airborne contaminants or microorganisms can be transferred from the unclean surfaces to the milk during the packaging process. Factors affecting the growth of microorganisms. The microbial growth in milk is influenced by several factors which will either promote or inhibit the growth of microorganisms. All these factors are important to ensure the safety and quality of milk. One of the major factors is temperature. Most microorganisms grow best at temperature between 20 degrees Celsius to 40 degrees Celsius. This is considered as a danger zone for bacterial growth because at these temperatures, pathogens and spoilage bacteria can grow rapidly. Refrigeration, that is storing the milk at temperature below 4 degrees Celsius, can reduce the growth of most bacteria and extend the shelf life of milk. Pasteurization or ultra-high temperature processing can also kill most of the harmful microorganisms and reduce the growth of spoilage bacteria. Another factor is pH. Fresh milk has a slightly acidic pH around 6.7 to 6.9. This pH is ideal for the growth of some bacteria but not ideal for the growth of other bacteria. So during fermentation, lactic acid bacteria converts the milk sugar lactose into lactic acid, lowering the pH of the milk. As the pH drops, it creates an unfavorable environment for many spoilage and pathogenic bacteria. For example, Salmonella and E. coli. So these bacteria cannot survive in that acidic environment. But some bacteria like Listeria monocytogenes can still survive in mildly acidic environment. Moisture content. Milk is a high moisture medium, making it an ideal environment for the growth of most microorganisms. The high moisture will facilitate the metabolism of microorganisms, allowing them to proliferate rapidly. But low moisture conditions such as drying or dehydration of milk, like in powdered milk, can reduce the moisture that is available for the microorganisms and thus effectively prevent microbial growth. Nutrient content. The milk is a rich source of nutrients including proteins, lipids, carbohydrates such as lactose, vitamins, minerals. All these nutrients supports microbial growth. This is also one of the reasons why the milk is susceptible to contamination if it is not handled properly. The milk sugar, that is lactose, is a key energy source for bacteria, particularly lactic acid bacteria. So these bacteria will ferment lactose and produce lactic acid. Oxygen levels. Aerobic bacteria like Pseudomonas species requires oxygen to grow. So these bacteria are more likely to multiply in milk which is exposed to air. That is, for example, milk which is left open. Anaerobic bacteria like Clostridium species do not require oxygen to grow. So they can survive in sealed environment and also under poor sanitary conditions. This can lead to the spoilage of milk and also production of gases like soaring and swelling of milk containers. Time is another important factor. The longer the milk gets exposed to favorable conditions like improper storage, high temperature, the more time the microorganisms are having to grow and multiply. Additives. Salt acts as a preservative by drawing out water from the microbial cells, creating a hypertonic environment and thereby inhibiting the growth of bacteria. Certain preservatives or antimicrobial agents can be added to the milk or dairy products to reduce microbial growth. For example, sodium benzoate and niacin can be used to prevent the growth of unwanted microorganisms in specific dairy products. Milk composition. Milk fat can also affect the growth of bacteria. Some spoilage bacteria prefers fat-rich environment, whereas some bacteria consider the fat content of the milk as an inhibitory factor. Now, proteins in milk like casein and whey proteins are also excellent nutrient sources for many bacteria. So, the breakdown of milk proteins during fermentation by lactic acid bacteria can lead to the production of desirable products like yogurt and cheese. But it can also support the growth of spoilage organisms if it is not controlled. So this is all about milk microbiology. I hope that the video was useful for everyone. Do subscribe the channel for more informative videos.